Without further hesitation, may I pass over to Dr. John Fernachez for your presentation for today.
Thank you for the kind introduction. Welcome everyone, and thank you for choosing to attend this webinar on Eurofins Discovery's new SH2 scan binding assay platform for the identification of small molecules that selectively bind to human SH2 domains. Through our experts, Eurofins Discovery can assist at the various stages of your drug discovery projects, providing services and products in the areas of synthetic chemistry, in vitro pharmacology, safety pharmacology, custom assays and products, in vivo services, translational phenotypic services, biotherapeutics, admitox, and integrated drug discovery. While the scope of today's presentation will focus on our in vitro profiling assays, be sure to reach out to our service representatives if you're interested in any of our other offerings for your drug development projects. First, I will be presenting a brief overview of Eurofins Discovery Lead Hunter Services' well-known KINOMEscan screening platform. I will then discuss how SH2 domains can be exploited as a novel drug target for kinases and other protein target classes. This will then lead to a discussion of our new SH2 scan competitive binding assay panel, which is based on the same technology as the KINOMEscan platform. I will show validation data for our kinase and STAT SH2 domain binding assays. I will then present an overview of our custom assay development services and end with a summary of the advantages associated with using the SH2 scan platform for your drug discovery program. This will be followed by a question and answer period. Eurofins Discovery Lead Hunter Services offers a collection of competitive binding assay panels for various target classes. In each competitive binding assay, 3 key components are present: the target protein tagged with DNA, a known ligand of the target protein immobilized on a solid support, and a test compound or solvent control. The DNA tagged protein of interest is incubated with a known ligand immobilized to the solid support. In the presence of a competitor test compound, the target protein is competed off the solid support. After washing of the beads and dilution of residual protein bound to the solid support, the amount of DNA tagged protein is quantified via an ultra-sensitive qPCR readout. In the presence of a competitor test compound, the final qPCR readout is expected to have a low signal. In the presence of a solvent control, very little protein is competed off the solid support. After washing of the beads, dilution of residual protein bound to the solid support, and qPCR quantification of the DNA tagged protein, the assay signal is expected to remain high. A dose response of a competitor compound can be tested using this assay, and an accurate thermodynamic dissociation constant for the competitor can be extracted from the resulting data. This competitive binding assay principle has been used to design our KINOMEscan, BROMOscan, BCL2 scan, E3 scan, and SH2 scan platforms. SH2 scan will be the subject of the latter part of this presentation. Kinases remain a key target class in drug discovery, and assays targeting kinases remain an important part of the Eurofins Discovery portfolio. Eurofins Discovery Lead Hunter Services is well known for their KINOMEscan platform, which allows for the discovery of new selective kinase active site inhibitors. Profiling of small molecules with this platform can quantitatively measure target selectivity across the kinome in a robust, high throughput, and highly accurate fashion. Numerous high-profile scientific publications have used data generated by KINOMEscan to support drug discovery efforts. The discovery of the second-generation selective and covalent BTK inhibitor Calquence by AstraZeneca was supported by data generated through the use of KINOMEscan. Using our platform, the compound was shown to have a greater selectivity and minimal off-target activity in a direct comparison to the first-generation inhibitor, ibrutinib. KINOMEscan has been further used to support IND packages, such as this example of APS-03118, a highly selective and potent next-generation RET inhibitor. In light of the successes achieved through the use of our KINOMEscan platform, Eurofins Discovery has further extended this technology to a new target class, SH2 domains, through our SH2 scan platform. There are 120 SH2 domains annotated in the human genome, and these protein-protein interaction modules are found in multiple functional protein classes, including kinases, transcription factors, E3 ligases, and adapter proteins. Dysregulation of the biological function of SH2 domain-containing proteins, such as through mutation or overexpression of the target gene product, can lead to pathological states, such as cancer and chronic inflammation. SH2 domains act as protein-protein interaction domains through binding of phosphotyrosine residues on target proteins. The selectivity of the interaction is imparted by the flanking amino acids surrounding the phosphotyrosine residue on the recognized protein. The function of the SH2 domain is context dependent. For example, an SH2 domain can act as a regulatory module for protein catalysis in one protein, or as a recruitment scaffold for other binding partners in another protein. Importantly, not all SH2 domain-containing proteins have enzymatic activity, and many of these proteins are emerging drug targets, such as transcription factors. As part of a case study, the first-generation kinase inhibitor imatinib was screened using the KINOMEscan platform against wild-type and the drug-resistant M351T mutant of ABL1 kinase. We detected an over 10-fold decrease in affinity of the compound for the mutant enzyme, which is consistent with the resistance phenotype observed for this drug in patients. One way of circumventing drug resistance is to develop drugs that target a novel site which is not impacted by acquired resistance mutations. The SH2 domain is one such possible site. We've currently released a collection of 26 kinase SH2 domain binding assays as part of the SH2 scan platform. In order to validate our kinase SH2 domain binding assays, we tested a semi-synthetic phosphopeptide from the scientific literature to determine the selectivity of this compound against our assembled SH2 domain assays. We observed an over 1,000-fold difference in selectivity for the kinase SH2 domain assays presented in the table on the right, demonstrating the ability of our assay to quantitatively assess binding affinities of ligands over a broad range of concentrations. Another important class of SH2 domain-containing proteins are the STAT transcription factors. STAT proteins play critical roles in canonical signaling pathways in cell growth and differentiation, and in inflammation. Deregulation of the biochemical functions of the STAT proteins have been associated with cancer and autoimmunity. Drug discovery for transcription factors is increasingly attracting attention, particularly in the context of innovations in covalent inhibitor chemistry and targeted protein degradation. The STAT SH2 domain binding assays and SH2 scan include 7 wild-type STAT SH2 domains, as well as 2 STAT3 mutant SH2 domains and 1 STAT5b mutant SH2 domain. To validate the STAT SH2 domain assays, we utilize compounds selected from the scientific literature, which have been shown to be selective for their specific targets. The STAT3 SH2 domain targeting small molecule SI109 and its related bifunctional protein degrader SD36 were profiled against the STAT panel. In addition, the STAT5B SH2 domain inhibitor STAFIB1 was profiled in the STAT assays. Sample curves for the binding of SD36, SI109, and STAFIB1 are shown on this slide, while selectivity data for the 3 compounds used to validate our panel are shown in the table. Rank order and selectivity of the compounds tested tracked closely to the affinity values published in the scientific literature. The exquisite sensitivity of the qPCR readout of the assay system allows for the use of low picomolar amounts of protein in our assays. As such, our assays do not approach the tight binding limit even for compounds with picomolar affinity, allowing for the measurement of a broad range of KDs during compound profiling. Furthermore, during assay development, the bait loading of the immobilized capture ligand on the solid support is optimized so as to not have an effect on the measured KD of the competitor. As such, true thermodynamic KD values are reported for control and test compound competitors. Currently, Eurofins Discovery offers 26 kinase SH2 domain and 10 STAT SH2 domain binding assays in KD elect or dose response format. Compounds can be profiled through the complete collection of assays, or assays may be selected on an ad hoc basis to form a custom panel. We are planning to offer single dose compound screening in 2024 for SH2 scan. In addition to off-the-shelf assays available in our screening panels, we also offer custom assay development services on a case-by-case basis. Here is a flowchart for the general workflow for a custom assay development project. The initial stages, which involve cloning and synthesis of the immobilized affinity ligand and the control competitor, as well as the assay proof of concept and optimization, take generally 6 to 8 weeks, while compound screening, hit confirmation, and potency determination is performed in the 2 to 3 weeks following assay onboarding. There are major advantages in using the SH2 scan binding assay platform for drug discovery programs. We plan on launching over 100 wild-type SH2 domain binding assays for this platform throughout the end of 2023 and into 2024, which will allow for the screening of small molecules across the target class for selectivity evaluation. Collected data are accurate, precise, and reproducible, and there is a high sensitivity and a broad dynamic range in KD measurements for competitor compounds. We have internal reference compounds that we have characterized for binding affinity, and all assays are run in parallel on a single platform, allowing for direct comparison of data between targets. This is the largest binding assay panel available on a single technology platform for human SH2 domains, all without having issues of compound interference in the assay readout. Assays are performed in a high throughput 384 well format, allowing for library profiling. We offer rapid turnaround for compound screening of 5 days for weekly submissions and 20 days for library screens. In summary, I've presented an overview of the new SH2 scan binding assay platform offered by Eurofins Discovery, which can be leveraged for the discovery of selective small molecule inhibitors targeting SH2 domains. We provided validation data for this panel using small molecules and a bifunctional degrader from the scientific literature, and recapitulated published rank order and selectivity data for these compounds. The SH2 scan binding assay platform can be used in parallel with our E3 scan platform for the design of bifunctional degraders targeting SH2 domain containing proteins. Functional characterization of small molecule inhibitors or protein degraders of SH2 domain containing proteins identified using SH2 scan can be performed using our Sprinter, InCell, and Biomap platforms. Furthermore, we offer custom assay development for your target of interest using the same technology that underpins our other competitive binding assay platforms. With that, I would like to thank you all for attending, and I would be happy to take questions at this time.
Great, thank you, Dr. John Vernatchez, for your excellent presentation. We now have the pleasure to introduce our question and answer session, where today's speakers will answer all of your questions. So don't forget, you can still submit your questions using the questions panel in the menu on the right. So if I invite Jean to the screen, please. Our first question is, what is the turnaround time for custom assay development for this assay?
Right. Thank you very much, Chloe. The turnaround time for custom assay development, as described in the presentation, is 4 to 6 weeks for clone generation and assay proof of principle, so to establish the assay window, and then 2 to 4 weeks to do assay optimization, where we determine the correct bait load gradient and the correct top concentration for the dose response of the test competitors that are used in the assay. We also offer within that period, in that particular milestone, the option for the client to test up to 4 of their own compounds within the assay to validate it before it moves to production. And then within production, as I mentioned in the presentation, it's with about a 2 to 3 week turnaround time for the library screens.
Great, thank you. So we have another question here, which is, will you develop a custom assay that is not in your current menu and terms?
Absolutely. So usually what happens is we speak with the client and we assess the literature that's available around the specific target in terms of construct availability and either peptide or small molecule ligands that can be used as capture ligands or as competitors for the assay. Given that information, if there is sufficient information available in the scientific literature, then we decide to go forward with that and we assign how much risk there is in the particular project. And we have that all written out and agreed upon with the client before moving on to the assay development process.
Great, thank you. So our next question is, can this assay detect compounds that bind to another site on the SH2 domain than the one that binds the capture ligand on the bead?
Theoretically, yes, it is possible. You can detect compounds that bind to another site on the SH2 domain insofar as that they need to cause a conformational change and a disengagement of the capture ligand from the protein of interest, the SH2 domain in this particular case. It's more likely you're going to find a direct competitor where the binding sites overlap, but it is theoretically possible for you to find in kind of more extrema cases allosteric compounds, but mostly you're going to be finding direct competitors.
Okay, thank you. So we have another question here, which is, can 2 very similar SH2 containing proteins discriminated with the same small molecule in the assay? I don't know.
Yes, yeah, it is possible. We have demonstrated in the slide deck the selectivity, for example, of SI109 and SD36 across the STAT transcription factor SH2 domains. These proteins have fairly similar binding pockets, and we were able to pick up the discrimination between those particular proteins in terms of the selectivity across the class. We saw, a difference of over 10,000, fold difference in terms of KD.
Okay, thank you. So for this assay, does the KD of the test compound dependent on the affinity of the capture ligand to the SH2 domain?
Right. As mentioned in the presentation, there was a mention in my presentation that we do a bait load gradient during the assay optimization process. This applies to all technology platforms that rely on the KINOMEscan basic technology, where upon doing the bait load gradient, we establish the concentration of the capture ligand on the bead at which the KD of the competitor, the test competitor for the assay, no longer changes with bait loading. And at that particular point, that is said to be the correct bait loading for the assay development. at which point the KD of the test compound is no longer affected by the affinity of the capture ligand for the target protein that's bound to the bead.
Great, thank you. We have another question here, which is there a cross-reactivity among the SH2 domains STAT proteins?
Cross-reactivity, yeah, well, I'm not quite understanding exactly, but there are compounds that we have found that are much more highly selective for certain STAT proteins than for others. Without getting into any kind of proprietary details, I have seen compounds that have gone down to the single picomolar in terms of KD.
Okay.
And we've had ligands that don't bind at all, up to 30 micromolar. Yeah.
Wonderful, thank you. Let's just look through some more questions. What are the other types of SH2 domain assays that you offer?
Good question. We are planning to offer, as I mentioned in the presentation, up to, we're hoping to be able to launch most of the entire class by the end of this year until next year. We have a good amount of assays approaching 100 that are currently in the process of being onboarded into production, and we're hoping to be able to launch these either by the end of the year or into the beginning of next year. And we are working to complete the rest of the class on the back end. So we're hoping to have a comprehensive panel so that people can do selectivity screening across the entirety of the target class.
Perfect. Someone is asking for the contact details for conducting Kinome screening.
Sure, absolutely. You can either reach out to myself or to the available available study directors on our website. We have the link to the email address to address that, but you can also contact me and I can put you in contact with the correct individuals that would be able to handle your project.
Great, thank you. Our next question is, a few years ago, the SH2s were considered undruggable. What changed in the past few years, and what publications motivated Eurofins to develop the SH2 scan?
Right, so a number of publications have come out recently, a number of projects that have been presented at recent conferences and drug discovery projects that have been publicly announced have come out, mostly targeting STAT transcription factors, for example. There have been a number of high-profile publications that one of which that covered SI109 and ST36 was another publication that came out from the same group recently within the last year, also on selective degraders for STAT5. Mostly the major kind of developments that changed in recent years is the reemergence of covalent chemistry as a viable option for targeting these particular proteins, as well as targeted protein degradation, which has also regenerated an interest in these quote unquote undruggable targets that have previously kind of been discarded, being revisited right now at the current time.
Perfect, thanks. So our next question is, does the Eurofins library contain phosphotyrosine mimetics or analogs? Is there a specific library subset that will be used for SH2 domains?
Right, so what I can say is that most of our capture ligands are based on either phosphotyrosine, natural phosphotyrosine containing peptide ligands, or small molecules that have mimetic moieties that mimic the phosphotyrosine motif that is used by these protein-protein interaction modules to naturally bind to their substrates. We have some data sets that we will be hopefully sharing in the next while to be able to show this particular information, but that is currently being developed, and we hope to be able to share that in the coming months.
Great, thank you. Is there an assay for the SH2 domain binders in the E3 ligases? Can selectivity be achieved here too?
Yes, so we do have a number of assays where we have E3 ligases that have SH2 domains that are going to be also launched in the MAX panel we, without a kind of naming them all, we definitely have a number in the SOX E3 ligases that have SOX E3 ligase family that have SH2 domains as substrate recognition domains, and these are currently being onboarded into our MAX panel. So yes, you will be offering that subfamily of E3 ligases with their SH2 domain assays within this panel.
Great, thank you. Got a few more questions. When are you planning to release additional assays for the SH2 domain?
Right, so the idea would be to launch another batch by end of year, and then hopefully as many as possible to complete the 120 assays within the first quarter of next year.
Wonderful. And are the assays with full-length constructs or domains?
Right, so all of these assays currently have only the SH2 domain, the ones that have been launched, the 36 that I presented today, the 10 STAT assays and the 26 kinase. The exact length and nature of the construct is posted on our website for each individual assay, so you can actually check that to see and compare that to your own internal constructs if you're working on these as well. We have a couple of these assays which might have an additional domain, the ones that haven't been launched yet that we will be launching because it's required for protein stability and for ligand engagement. And I'm thinking mostly towards the SOX E3 ligases. So, but in the vast majority of cases, it is strictly SH2 domain isolated alone. But in certain corner cases where we were unable to get adequate, protein engagement with the ligand or protein folding, then we had to, add an additional subdomain on to basically stabilize the protein.
Perfect, thank you. We've just had another question in saying, what about ring E3 legacies?
Sure. So we are currently developing those in the context of our E3 scan platform. So we do have a few of these that are offered, and we are actively developing assays in that particular area as well. If you're interested in a specific ring E3 ligase, that is something you can contact me for, and we can look into the literature and see if it's feasible to develop it, if there are ligands, if there are known constructs. I didn't mention it necessarily today, but we are also involved in developing E3 ligase substrate recognition domain assays through our E3 scan platform. So that's another technology platform that we also are always adding to, and we're always interested to hear from clients as to what assays they'd like to have in that panel.
Great, thank you. Our next question is, what is the TAT for running the panel?
Sure. So as I mentioned in the presentation, we're looking at a 20-day TAT for running running a library, and depending on whether or not you're running the existing panels on a few compounds, we're looking at 10 business days for running, like for example the kinase panel. For library screens, it's a bit longer because there are more compounds, but yeah, for routine, a few compound KD runs on the full panel, we're looking at 10 business days.
Okay, thank you. I think we've got time for a couple more questions. So we have an internal library of compounds. Can we send these for screening?
Absolutely. So we screen both client-provided libraries, and we also have internal libraries as well that are also available for clients. So we have the compound management ability to basically handle both client-provided libraries and our own internal libraries, depending on what the client would like to have screened.
Thank you so much. And I think we have time for one more question. How does the assay work where targets have tandem SH2 domains?
Right, so we have two assays currently in the panel, the one for ZAP17 and for SIC, which contain two tandem SH2 domain modules because they interact with their natural substrates via the two SH2 domain binding mechanism, where they interact with two closely located phosphatidylserine residues on their target substrates. We are planning to, in the new year, also develop isolated SH2 domain assays for either the C or the N terminal SH2 domain for both of those particular targets. So stay tuned for that. We'll be launching those also. We intend to develop those as well.
Great. Well, thank you very much. Unfortunately, that's all we have time for today, but please join me in thanking Dr. Jean Benitez for his time today.